So where have we seen this before? This is simply our glycolysis pathway. Now we've already taken a look at glycolysis and in part of how it's regulated. It's pretty logical to understand why the end product, such as ATP, is going to inhibit the glycolysis pathway. Remember, glycolysis is used to make ATP, so if we have a lot, it's going to inhibit this process. However, it's not as intuitive to think of why fructose 2,6-bisphosphate is going to alter the glycolysis pathway. So let's go over that. Now, you need to think of fructose 2,6-bisphosphate as a regulation molecule, or kind of a side process that's going to be going alongside of this glycolysis pathway. So here, let's introduce some enzymes. We see our phosphofructokinase 2 enzyme, which is going to make this molecule, this regulation molecule. And then we also have another enzyme, our fructose bisphosphatase 2, which is going to degrade it. So we kind of have a build and degrade process that's going alongside or concurrent with our glycolysis pathway. Now, the fructose bisphosphatase 2 and the phosphofructokinase 2 are actually part of the same protein complex. However, they respond in opposite ways due to phosphorylation. So if it's phosphorylated, it'll act in a way, and if it's dephosphorylated, it'll act in a certain way. Now, that's going to be regulated by protein kinase A. And we'll talk about protein kinase A later on in the pharmacology section. Let's compare the regulation of this enzyme in the fed and in the fasting state. Because remember, in the fed and the fasting state, our metabolic reactions act differently. In the fasting state, there's high glucagon levels, and this increases the amount of cyclic AMP in the cell through a G protein system. Now, this in turn is going to increase the amount of protein kinase A activity, which goes on to phosphorylate fructose bisphosphatase 2. So we're going to phosphorylate it here, and we're going to activate it. So notice how fructose bisphosphatase 2 breaks down our fructose 2,6-bisphosphate molecule. We're going to get rid of it. However, at the same time, we're also going to inhibit our phosphofructokinase 2 enzyme because we don't want to be making it, we want to be breaking it down. So in the fasting state, we're going to increase protein kinase A activity, and that will affect our side regulation here. Now, this means that you're going to be converting fructose 2,6-bisphosphate into fructose 6-phosphate and inhibiting the opposite reaction, which in turn is going to allow gluconeogenesis to proceed to replenish the blood glucose levels, and it just makes sense if you're in the fasting state. You want to replenish your blood glucose levels. Now, what do you predict in the fed state? In the fed state, high insulin levels decrease the amount of cyclic AMP and therefore decrease protein kinase A activity. So fructose bisphosphatase 2 will be dephosphorylated. Remember, it was phosphorylated in the fasting state, but in the fed state, we'll want to remove that phosphorylation. So therefore, we decrease fructose bisphosphatase 2 activity and increase phosphofructokinase 2 activity. So keep in mind that phosphofructokinase 1 is going to be the rate-limiting step for glycolysis to proceed. So in this step in glycolysis, it's going to be important to regulate this enzyme more than others. Remember, I said that our fructose 2,6-bisphosphate was kind of a side reaction. However, earlier we talked about the molecules that regulated this rate-limiting enzyme step. So here we see that it's being made and degraded based on whether you're fed or fasting. Now, over here is just a figure that we've seen before. Remember, low AMP levels, or high AMP levels can stimulate phosphofructokinase 1 because there's not enough ATP in the cell. But also I said fructose 2,6-bisphosphate affected it, and now you kind of know why. It's a side reaction off of glycolysis that's meant to regulate our rate-limiting enzyme. Now, that brings us to a flash quiz. A hard-working medical student on her surgery clerkship skips lunch to go to the OR. How does this affect the action of fructose bisphosphatase 2 and phosphofructokinase 2? So we're kind of integrating what we just learned into a possible test question. How is skipping lunch or going into the fasting state affect these two enzymes? Think you know it? Now, in the fasting state, an increased glucagon level increases your cyclic AMP levels. So let's write this out. Fasting is going to be increased glucagon, which is going to in turn be increased cyclic AMP levels, which in turn is going to increase protein kinase A activity, which is going to ultimately go on and phosphorylate. Which one is it going to phosphorylate? Do you remember? It's going to be our fructose bisphosphatase 2. That's the enzyme that's going to break down that fructose 2,6-bisphosphate. So it's going to phosphorylate fructose bisphosphatase 2, and then it's also going to inhibit our phosphofructokinase 2. So therefore, we're going to decrease levels of fructose 2,6-bisphosphate. That's going to be our take-home message here. We're going to decrease levels. Therefore, we're going to increase gluconeogenesis. Remember, she skipped lunch. She needs to make some glucose that'll be available for the blood. We need to go through gluconeogenesis. Do you remember what I said what the pyruvate dehydrogenase complex does? Remember, the pyruvate dehydrogenase complex here links glycolysis in the cytoplasm of the cell to the TCA cycle, which occurs in the mitochondria of the cell. Now, the net reaction converts pyruvate, NAD, and coenzyme A into acetyl-CoA, CO2, and NADH. Now let's look at this in just a little different way. Here we see kind of the same thing, just in a different form. So here's our net reaction. Here's kind of our reaction process. So here we see the pyruvate dehydrogenase creating an activated carrier. Now notice how we talked about our carriers earlier. The NADH and the NAD are electron carriers. So we're shuffling some electrons around, creating a reduced carrier, which will be used later on. Now, the cofactors needed for this reaction include the following. Pyrophosphate, which is simply two phosphate groups, and this is supplied via vitamin B1 in the form of thiamine pyrophosphate. We also need an FAD, which is vitamin B2. We also need an NAD, B3. We need coenzyme A, B5, look at all these B vitamins being used for this pyruvate dehydrogenase complex. And then also we need a lipoic acid thrown in there as well. Now, on a side note, this last one, the lipoic acid, is inhibited by arsenic and arsenic poisoning, which will cause vomiting, rice water stools, and garlic breath. Arsenic poisoning. If you're exercising, you probably want to be undergoing aerobic metabolism, and therefore you want the pyruvate dehydrogenase complex to be active, because glycolysis gets us a few ATP. Remember, we got a net of 2 ATP. The TCA cycle is where it's really at, though. This is where we get all of the rest of the ATP. But it doesn't produce it in the form of ATP. It's going to produce most of it in the form of activated carriers, which will go on later through oxidative phosphorylation and the electron transport chain. So everything's kind of linked. We need to link glycolysis to our TCA cycle where we can create a lot of ATP. During exercise, the ratio of NAD to NADH decreases since you're consuming all of that NAD to make a more reduced form of NADH, which will then go on to the mitochondria, exchange the electrons to make lots of ATP. Other changes during exercise include you'll have ADP levels increase, and then also calcium levels, and these together are going to activate the pyruvate dehydrogenase complex. So these are going to activate it. Now, the pyruvate dehydrogenase enzyme is going to be inhibited by substances such as ATP, because remember, our pyruvate dehydrogenase is going to link our glycolysis to the TCA cycle where we make tons of ATP. So if we have a lot, it makes sense that it's going to inhibit. Also, things that will inhibit are high levels of NADH. Remember, that's a product. Products tend to provide negative inhibition. And lastly, high levels of acetyl-CoA, which is another product, is going to contribute to the inhibition of the pyruvate dehydrogenase enzyme. So we see some negative feedback by these products. And then ATP, remember, this whole thing is connected to the process of making lots of ATP. If we have lots of ATP, we don't need to run this pyruvate dehydrogenase complex. So these molecules right here have negative inhibition or negative feedback on the pyruvate dehydrogenase. It just makes sense. What enzymes in the TCA cycle use the same set of cofactors that we see here? The cofactors for the pyruvate dehydrogenase complex show up again. These aren't just an isolated, pure memorization. We need to memorize these because we'll see them again in multiple different situations. We'll see these cofactors needed again when we talk about alpha-ketoglutarate. Now that's one of the enzymes in the TCA cycle. The alpha-ketoglutarate dehydrogenase complex is going to convert alpha-ketoglutarate into succinyl-CoA. And that's going to be in the TCA cycle that we see here. So our alpha-ketoglutarate enzyme dehydrogenase complex, so alpha-ketoglutarate dehydrogenase complex. This is our enzyme that's going to convert our alpha-ketoglutarate to succinyl-CoA, going to use these same cofactors here. Now, if you can remember this one, you can remember the alpha-ketoglutarate dehydrogenase cofactors as well, since they use the same cofactors, and they both have the same dehydrogenase complex, which decarboxylates their substrates. Let's now move on to a clinical application of the pyruvate dehydrogenase complex. So, a PDH complex deficiency is going to be a serious problem for all cells with a mitochondria, since it prevents the products of glycolysis from entering the TCA cycle and therefore prevents aerobic metabolism. Now, this creates a buildup of pyruvate in substrates, increasing the amount of pyruvate in alanine and therefore shunting it via anaerobic metabolism to create lactic acidosis. Now, this PDH deficiency can be either congenital or acquired. Now, why do you suspect alcoholics can present with an acquired pyruvate dehydrogenase deficiency? So this would be a form of acquired deficiency. Why do you suspect alcoholics may present with an acquired PDH complex deficiency? That's because alcoholics typically are nutrient deficient, especially in the vitamin B1. Now, do you remember what vitamin B1 is? That's going to be your thiamine. So, since they're deficient in vitamin B1, do you remember what the cofactors were that were required for the PDH deficiency? Yeah, that included vitamin B1. So, boards love testing this concept, so don't be surprised if you don't see it on test day. Clinically, this is going to manifest as neurologic deficits or lactic acidosis. Remember, we're going to have a buildup of lactate. Now, it's going to be treated by increasing the intake of ketogenic nutrients, such as a high-fat diet or a diet high in the ketogenic amino acids, which are going to be lysine and leucine. Remember, this can also be congenital. So in alcoholics, you're going to give vitamin B1. But if it's a congenital issue where you're missing or this enzyme is not working correctly, you're going to use ketogenic amino acids. Now, these nutrients do not have to go through the pyruvate intermediate to enter the TCA cycle. That's why we choose leucine and lysine high foods, because they bypass the PDH complex, and these go directly into the TCA cycle. So they're going to bypass our PDH entry here.